Biolog's revolutionary Gen 3 system is the first and only bacterial identification system to identify both gram-negative and gram-positive aerobic bacteria with a single universal test kit. This makes it the simplest of all to use. No longer is a gram stain required, nor any pre- or follow-on tests. In less than 1 minute, bacteria can be taken from an agar plate and set up for identification. Over 1,000 species can be identified. Biolog test kits employ a simple universal single-color redox chemistry to detect reactions of bacterial cells. Each well is a different test, and an ID is run by simply adding a cell suspension and incubating. Over a period of hours, cells will generate energy in some of the wells, reducing the redox dye and forming a purple color in all positive wells. The arrangement of the 96 tests is shown here. On the left side of the panel are 71 carbon source utilization tests. On the right side are 23 chemical sensitivity assays. This slide shows a result with Stenotrophomonas multifila. This bacterium can metabolize a variety of sugars, a hexose phosphate, some amino acids, a hexose acid, and a variety of carboxylic acids, esters, and fatty acids. In chemical sensitivity assays on the right side of the panel, the top row shows the ability of the bacterium to tolerate acidic pH, pH 7, pH 6, and pH 5. This bacterium will tolerate pH 7 and pH 6, but pH 5 is too low. The next row has salt sensitivity, 1%, 4%, and 8%. 1% and 4% are tolerated, but 8% is too high. This bacterium is resistant to lactic acid and has strong reducing power. It is resistant to vancomycin and sensitive to nalidixic acid, all consistent with it being a gram-negative bacterium. Here we show results with 4 very different bacteria. On the left are 2 gram-negative bacteria, Moraxella cataralis and Acinetobacter baumannii. On the right are 2 gram-positive bacteria, Corynebacterium pseudodiphtheriticum and Streptococcus pneumoniae. These bacteria are extremely different, but they all react very well in this universal test panel. Here you can see, for example, in the Streptococcus, that the carbon sources are primarily positive for sugars, but negative for acids, whereas Acinetobacter gives the opposite results. It primarily uses acids rather than sugars. Also, you can see, highlighted in the blue box, that the Acinetobacter baumannii is very resistant to inhibitory chemicals, whereas Moraxella is very sensitive. Here we show 4 other examples, 2 gram-negatives on the left, Stenotrophomonas multifila and Sphingomonas palsamobile, 2 gram-positives on the right, Staph aureus, and a spore-forming gram-positive, Pennibacillus polymixa. Even so-called non-reactive bacteria give clear positive reaction patterns and very good identifications in the Biolog Gen 3 panel. Here we show some examples of Campnocytophaga, Neisseria, Actinobacillus, and Moraxella. The Gen 3 system has a number of important features and advantages. As already indicated, it identifies both gram-negative and gram-positive aerobic bacteria in a single universal panel. The 96 tests of the panel were selected after evaluating over 1,900 potential tests. This is the simplest and easiest bacterial identification system available. No gram stains are needed, no pre-tests or follow-on tests are needed. We have the largest database of any test kit system by a factor of 3. We use a universal temperature of 33 degrees Celsius to incubate and identify the cells. At this temperature, we get the maximum species coverage at a single temperature. Very few bacterial cells are required to run an identification. An optical density of only 0.01 is needed, and the microplate setup time is only 1 minute. The new Biolog inoculator swab is used to touch the surface of an agar plate and remove bacteria for inoculation. It will automatically pick up the right number of cells. If you have a fast-growing bacterium, you touch the swab to a single colony. For a medium-growing bacterium, such as a staph, you touch to a cluster of 3 to 4 colonies. And for a slow-growing bacterium, you would touch it to the area of confluent growth. The swab will pick up the amount of cells needed to give an inoculum density of 0.01. The cells are then suspended in inoculating fluid. The Biolog turbidimeter can be used to verify the correct cell density. The cells are then pipetted in a single pass using the Biolog pipetter and then loaded for incubation and identification. Biolog offers 3 levels of systems: a fully automated OmniLog system, a semi-automated microstation system, or the panels can be read by eye using our manual system. The 1,000 plus species cover virtually all human pathogens and also major animal and plant pathogens, as well as commonly encountered environmental species. Biolog's database is much larger than databases found in test kits typically designed for hospital use. Here we show a comparison of the species we can identify compared to the new Vitek 2 system. In addition to aerobic bacteria, we can identify a lot more anaerobic bacteria, yeasts, and even filamentous fungi. Biolog has developed test kits for microbial use for over 20 years. These test kits have always been designed to address all fields of microbiology. We have major users of our systems in fields such as industrial quality control, veterinary microbiology, environmental microbiology, research and education, ecology, agriculture, food and water testing, and reference testing. In conclusion, the Generation 3 system is an ideal system for microbial identification. It is a truly modern and fully updated phenotypic testing system. Phenotypic testing systems have advantages over molecular systems in that they provide not only an identification, but also useful biological information about the various isolates being tested. The capabilities of the system also include fungal identification and characterization. Biolog also offers a phenotype microarray technology for very detailed cell characterization. We have different size systems for different size laboratories. The combination system that includes both the microbial identification and the phenotype microarray technologies is ideal for core laboratories. Biolog provides much more than just a microbial identification system. We offer scientists a uniquely powerful technology platform for a wide range of cell phenotyping and identification applications. This encompasses not only bacterial and fungal cells, but even mammalian cells. It is an ideal system for a wide range of applications.